Hi guys, I welcome you all to the next lecture in our anthropology series. So we were doing human genetics and today we are going to complete the human genetics. Two topics were left. One is recombinant DNA technology and one is immunological methods, right? So let's start with recombinant DNA technology. As the word only suggests, recombinant, ठीक है, recombinant, that means they are talking about certain combinations and recombinations, right? Recombinations, okay. So not sorry, it's not recombinations, it is recombinant, but I'm just telling you कि बात किसकी हो रही है. We are talking here about something recombination, right? That means we have, for example, we have a this as a, you can say, a cell, right? But we don't want this part, ठीक है, of the cell or the DNA, right? So what we are doing is we are removing it, or instead we are putting in some other part of it, ठीक है. So this will become recombinant. Now what is DNA? The next word is DNA. So we have talked about DNA also, that DNA is the you can say is where all the genetic information is coded, right? So this is part of the you can say the genes and the chromosomes में there is the DNA and all the information, all the genetic information is encoded in the DNAs. So आपको DNA का भी पता होना चाहिए कि DNA एक ऐसे double helical structure होता है. It's a double helical structure and it has different bonds, ठीक है. So this is, you can say, a structure of DNA, ठीक है, and it is made up of different you can say enzymes जिसमें adenine, guanine, uracil, all of this is there. But these are the things that nobody is going to ask you ki what is DNA, what is it made up of. So it is made up of these bonds of uracil, adenine, guanine. So this is something which is okay, good for your understanding, but this nobody is going to ask, and that is why I was not telling it to you, ठीक है. But you should know what is DNA and where is it found, what is the reason for it. So we got it that DNA is the thing in which our genetic information is coded. For example, how much fat the body is going to deposit, ठीक है. The genes regarding your color, ठीक है. The genes regarding your intelligence, your height, your eye color, your body color. So these are different kind of traits that you have as a human being, and there are so many DNA that are there in the chromosomes. So recombination में क्या होता है? Basically when we are doing the study of genetics is all about, I'll tell you, if I have to summarize what is genetics is the study of genes. इतना तो genetics के नाम से समझ आ रहा है that it is the study of genes. But what about the study of genes? पहली चीज, we are trying to understand what is our genes, what are they made up of, what kind of information is coded in it. So some kind of a analysis we are trying to do. और इसमें we are studying about the evolution, ठीक है. जैसे फॉर एग्जाम्पल हम बोलते हैं कि टुडे 2 to 3% ऑफ नियंडरथल जीन्स आर प्रेजेंट इन some European population, right? So what is this? We have studied, we have analyzed the genetics, right? And based on that we have come up with certain conclusions. So एक part हो गया analysis. दूसरा part हो गया if you have inherited certain diseases, ठीक है. For example, you have inherited certain genetic disorders. So उन diseases का study, ठीक है. How to prevent those diseases, right? So prevention का, how do you diagnose that disease, right? Diagnosis आ जाएगा, your prevention will come, right? So your cure, how to cure them, or what vaccines you have to make, right? What treatments you have to do. So all this is what is contained in genetics. So this is all that we have as a, you can say, subject matter in genetics. So recombinant DNA technology जो है ना, that is one such way in which what we are doing, we are studying the DNA and we are trying to make certain new recombinations of DNA. Why do we need to do this? This is done because I'll tell you, recombinant, okay. Okay, so why we are doing, for example, let us suppose that this is a cell and I'll tell you that this part of my part of the cell is not working properly, so this part is defected. ठीक है, I have I have certain problem, so this part of the gene is defected. So now what can we do? For example, just think that this is a genetic disorder. So that means if I have it, it might be present in my future population also. So इसका क्या मतलब है that if there are genetic disorders, they will persist in the population. So now how can we correct it? What we do in recombinant DNA is we can, I'll just tell you a very crude way and then we'll go to the full explanation of it. What we'll do, we'll try and remove this part of the gene, and we'll try to put another part in the place of that gene which is not defected. So आपको समझ आया? What we are doing, there was a defected part in the gene, right? So what we do, we cut it and we do it through scissor, and this is not the scissor that we have at home. These are called genetic scissors. ठीक है, we'll take this part out. ठीक है, and in place of that we'll put a part that is not defected or which is healthy or functioning, and we'll put that part and that gene will become healthy gene again. So this is one way. और इससे हम क्या कर सकते हैं? We can treat a lot of diseases. ठीक है, this way हम treat करें diseases को, and this comes under the part of gene therapy. Therapy क्या हो गया? You are trying to cure something. So gene therapy में you try and treat the genes जो unhealthy है, not working है, semi functioning है, उनकी जगह आप उनको healthy बना देते हो. So this is gene therapy is one thing and one application of recombinant DNA. Similarly, for example, if I take example of BT cotton. What is BT cotton? BT cotton is you can say cotton as a plant is a fragile plant. That means it can be attacked by various pesticides, herbicides, right? So in recombinant technology or in BT cotton, उसमें हम क्या करते हैं? We are trying to introduce certain elements or certain genes that are resistant. For example, हमको pesticide resistant करना है, ठीक है? We want to make it pesticide resistance. So what we'll do is we will insert a gene that will make it resistant to the pesticides. So that becomes another kind of recombinant DNA technology, and agriculture में it has a revolutionary role to play. अभी हमारा BT mustard is going on and government has given, GAEC has given approval to BT mustard, so it becomes important. So agriculture हो, human diseases का point हो, the recombinant DNA technology has been revolutionary in many ways. ठीक है ना? So now let's see the explanation. So basically क्या होता है? We are trying to introduce a desired gene into the DNA of another species. We have a within species भी कर सकते हैं. It could be within species, it could be within two different kind of species. So it can be both ways. And इसमें what we are trying to do, sometimes we are adding new genes. ऐसा हो सकता है ना? जैसे pesticide resistant gene we added, that was a new gene. Sometimes we are trying to delete a old gene and in place of that replace it with the new. So इसमें we can do additions, subtractions, replacements, right? So all of this we can do. And how can we replace one gene from one organism to the other? It is because many genes in the humans or so to say prokaryotes and eukaryotes are similar in structure and function. अभी prokaryote और eukaryote क्या होता है? Prokaryote you can say primitive organisms, right? जो very simple organisms होते हैं, पुराने paramecium या वैसे टाइप के simple and primitive kind of. Eukaryote हो गए complex organisms, ठीक है, like humans, ठीक है, eukaryote हो गए. So many organisms in our environment have similar structures of genes, and not only similar structure of gene but similar functions of gene. So that is why it has made possible to do intra-species. For example, so this has made it very easy, right? सो इसमें हम ऐसे डिफरेंट चीज़ें कर सकते हैं, राइट. सो दिस इज़ वन थिंग. अब इसमें व्हाट आर द स्टेप्स, रिकॉम्बिनेंट डीएनए टेक्नोलॉजी का स्टेप्स क्या है. यू फर्स्ट, ओके लेट मी शो यू द पिक्चर एंड देन यू विल अंडरस्टैंड इट क्लियरली. नाउ फॉर एग्जाम्पल दिस इज अ जीन, ठीक है, ये एक जीन था, and this part of the gene we required, okay let me change the color, this part of the gene was required to us, ठीक है. So what we do, we take a restriction enzyme and we try and cut. So restriction enzyme हो गया scissor, it acts as a scissor, ठीक है, जो cut करता है. हमको इतना पार्ट चाहिए, we want this much part of it. So what we are doing through a restriction enzyme, enzyme ही ऐसी चीज़ है that is going to cut. It is not going to be the physical scissor that is going to cut. Enzyme होता है which acts as a scissor. Okay, so it's a restriction enzyme that will cut. Now we have taken that strand that we require, ठीक है, and now this is another gene, ठीक है, where we need to put this. So now we, with the help of ligase, another enzyme, ठीक है, now we put it back, as in put it in the new gene or the required gene. So we, after that we culture it, that means अब हमने बना लिया ना, so we have to put it back, insert it back into the cell. तो हमने इसको, we have put it back and then it will multiply on its own, because now it has become the part of the genetic material, so it will regenerate, right. So this is the whole process. So now you can see what we have written is that first we are going to isolate the genetic material that we require. Second step, we are going to cut it through the restriction enzymes. Then ligation, another enzyme you can say, which acts as a which acts as a glue, ठीक है, उसको paste भी तो करना है ना वापस. You have cut it, so now you have to paste it back to the next gene, right. So you put it back and then insert it into the host. होस्ट एज इन जिनसे आपने वो सेल, जिनका जीन आपने निकाला था, नाउ यू हैव टू पुट इट बैक इनटू हिम ना, सो दैट इज द होस्ट, ठीक है. सो यू इंसर्ट इट बैक एंड इट हैज़ अ वाइड वेराइटी ऑफ एप्लीकेशन because it is used in gene therapy. That means I've told you there are genetic disorders, there are genetic defects, you want to correct it, so उसमें use होता है. and इसमें eugenics का concept भी आ जाता है. But eugenics we are not going to cover it here, we are going to cover it in the next lectures. Gene therapy में use हो गया, you know genetic disorders हैं, to correct it वहाँ use हो जाएगा. Then there are diagnosis, many diseases can be, first you have to diagnose them also ना. So ELISA is one such mechanism जिससे you try and note down, ठीक है ना. तो ये भी recombinant का एक वो है, आपको पहले डिजीज को चेक भी तो करना है. So with the help of this you are going to check it. Then its application are there in agriculture, I've told you, ठीक है ना. So there are so many examples, BT cotton is there, flower saver tomatoes are there, golden rice are there. So in different things you are required to protect them from different things. For example, golden rice है, उसको आपको fortify करना है, so you need to add certain things, you need to put certain things into it. So with the help of recombinant technology you can do that. Then in in production of many enzymes like insulin, you can produce insulin, ठीक है. Other enzymes you can produce like growth hormones, ठीक है. So these all things are the, you can say, modern applications to it. So I hope you have understood, and you can make certain vaccinations also. So recombinant DNA technology के basis पे hepatitis B, foot and mouth diseases, these are certain diseases जिनकी vaccines have been made with the help of recombinant DNA technology. So I hope you are now clear with the idea of recombinant DNA technology. Now let's move to a very short topic, that is immunological methods. Now do not worry, because as I've told you, this 9.1 was just a gist of the topics that we are going to cover in the further lectures. So here I'm not elongating it, as in when in the next chapters it is required, we are going to cover it in detail. Okay, so immunological method is made from the word immune, ठीक है ना. So immune and immune system you can say. So what is immune system? Immune system is basically, you can say it deals with, largely deals with your different blood groups and your antibodies and antigens. Now what are antibodies and antigens? Your blood, your blood stream has certain antibodies and antigens which are activated at different times. So what happens is, for example, certain foreign agent enters your body. So there is an entry of a foreign agent. For example, जैसे हम कोविड के केस में बोलते हैं, what is COVID? COVID is a virus. So that's a foreign agent that enters your body. So it acts as a antigen. ठीक है ना? Because it is not, it is something which your body doesn't recognize, right? It is something, a foreign element to your body. So your body tries to fight it in its own way, and that is called the immune system. So immune system क्या होता है? Your body has a certain system, okay, and anything which is not something which your body recognizes, so your body tries to fight it, and that is called immune system. So जब आप बीमार होते हो, so what happens basically when you fall ill, what happens that your body has tried to fight it. For example, you get a cold, right? So when you get a cold, it is not that, see, there are millions of viruses and bacteria around us, right? But not every day we are falling ill, not every time we are not well, right? That is because your body and its immune system is working and stopping all of those bacteria, viruses and other foreign elements. But sometimes your body is not able to react or react as strongly, ठीक है? So that is the time you fall ill. So उसमें क्या होता है? Immunological methods में we are trying to study the immune system, right? So इसमें क्या होता है? We are trying to study the immune system, how the body fights against the foreign elements. So whenever your body sees a foreign element, which is in this case an antigen, so your body produces antibodies, ठीक है? So antigen के खिलाफ में क्या produce करेगी? Antibodies, ठीक है? These antibodies help the person to fight against invasion by different microorganisms, right? So when we are studying the immune system, how the body reacts, how the body sees a foreign element, how it makes antibodies, this is all which comes in the immunological methods. Now immunological methods में blood type incompatibility पढ़ते हैं हम. जैसे for example, what is blood type incompatibility? Basically we know there are three systems, ABO. ABO blood groups होते हैं. That means there is either your blood group will be A, B, AB or O. Sorry, A, B and O. So these are the different kind of blood groups that we have. For example, now if I give a person A the blood of B, a person who has B blood group, so what will happen? This blood person who has blood group A, if I give him blood group B blood, so what will happen? That body will not recognize this blood because it is a foreign agent, or you can say in this case antigen, right? Antigen हो गया ना उसके लिए. So it will not recognize. So that is why a person who has a A blood group, he has to be given A blood only. And not only that, उसमें भी और बहुत सारे details आते हैं, उसके बाद check करने के बाद ही blood दिया जाता है. Because in other cases the blood it does not suit, so वो incompatibility आ जाती है. So incompatibility क्यों है? Because your body does not recognize it. It thinks that it is a antigen, and then it starts to work against that, start making antibodies, and the person falls ill and all of that. Then transplant rejections, ठीक है ना? Now you can't transplant anybody's organs like that. There should be वहाँ पे match होना चाहिए, that organ should be matched to the other person that you're giving. So to जैसे इसमें how people transplant and give each other जो भी organs होते हैं, so उसके लिए you need to see whether that is compatible with the person that you're giving, ठीक है ना? So ये सब आपके immunological methods में आते हैं. Basically आप foreign elements you're introducing into somebody's body, so it has to suit that person, right? You can't give anything to anybody, right? So that has to suit, and that is basically that we are studying in the immunological method. Because the moment you give somebody some foreign element, the body starts reacting to it. Then इसमें we study cell mediated type of immune reactions. हमारी body basically it makes two types of you can say immunity. One is T cell mediated and one is B cell mediated, ठीक है? Now what are these different kind of immunity? You need not go very deep into it, बस ये याद रख सकते हो कि T cell is a more of a long term kind of a immunity, or B cell you can say is derived from T cell and it's a short term kind of immunity. So for example if you take a certain medicine, ठीक है, तो वो आपको एक B cell type of immunity दे सकती है, right? But if your body produces some kind of a B cell, body में भी produce होती है, I'm not saying that it's not produced in the body, it is produced in the body only, but short term के लिए, right? It's short term kind of. T cell long term हो गई, ठीक है. So these all things we are studying in the immunological methods. And then there are immunodeficiency disorders भी हैं, जैसे कि हमने genetic disorders पढ़े थे ना, वैसे ही हमारी sometimes in our body there are immunodeficiency disorders also, so we study that as well. So now this completes your chapter 9.1, and tomorrow we are going to start with 9.2. Any doubts you can post in the comment section below, but I think these topics this much will be enough for you, because for example immunological methods, no question is going to come on immunological methods as such, so it will be a part of a bigger question. So I think this much information will be sufficient for you, but if you want to ask anything else, I'll be happy to answer you. So guys, please do like, share and subscribe. Thank you.